rest of the way through 12 because I think that's the way we should handle it. All right, so the next question we need to deal with from chapter 12 is this one. What role do chromosomes play in inheritance? So we know a little bit about chromosomes. I mean, I don't mean as humans. I mean as like what we've covered in this class. We know a little bit about chromosomes. And so then there's a question of, okay, well, what role do they actually play in inheritance? And as we look at a cell, so this is a cell that is going to go through meiosis. For most animals, and really for most organisms, most eukaryotes, this organism is going to be diploid. It's going to have 2 copies of every chromosome. And we're like, well, this is kind of interesting. That sounds a little bit like Mendel's ideas, where for every character you had 2 different, predictors or determiners, 2 different factors to determine that character. And then so here we've gone through where we're at metaphase 1, and we've got what we would call independent assortment going on, right? Where we can line up all of our maternal on the same side and all paternal on the same side, or we can line them up a little bit differently, right? And so this is a relatively simple situation where we only have 2 chromosomes, and then each chromosome has 2 copies. And then so after that meiotic division is done, after anaphase 1, we end up with basically 4 different types of nuclei. Not 4 different nuclei, because after meiosis 1 we only have 2, but there are 4 different types possible, right? And so one that has both paternal, so this would be a paternal gamete, okay? One that's basically matching the paternal contribution to you. This would be a maternal gamete. And these 2 are mixes. These 2 are gametes that haven't existed before, right? Because they're part from your mom and part from your dad, or if we're dealing with something that's not human, part from their mom and part from their dad. And then so after our second meiotic division, we have gametes that are now haploid. So instead of having 2 sister chromatids for each chromosome, now those split, and we have those into different places. All right? Now, in this situation, it had your 2 factors on separate chromosomes. They don't have to be. They could be on the same chromosome. But regardless, as we're looking into here, every gamete only has 1 version of that gene, right? So this gamete has 1 version of the R gene and happens to have the dominant allele. It has 1 version of the Y gene, also has the dominant allele. And you look back to the dad, and the dad was dominant for both, right? And then these gametes here still only have 1 copy of each gene and only have 1 version of that. Now it's starting to look a lot like what Mendel was saying, right? That the 2 different versions of any particular character segregate from each other during gamete formation. And here's the chromosome showing how that happens. And so here's showing you basically homologous chromosomes. And the reason why, how can we tell that these are homologous chromosomes and not sister chromatids? They're not connected, that's one clue. Although you could say, well, during anaphase 2, the sister chromatids are not connected either.
They don't have the same.
Yeah, they don't have the same version of the gene. Sister chromatids should be genetic copies of each other, right? Perfect copies. Unless, of course, there's a mistake that's made. But these have 2 separate alleles. So here we have homologous chromosomes showing you the 2 different alleles for that gene, or the 2 different traits for any particular character. Showing, using chromosomes, how basically the law of segregation works. So how do chromosomes work in inheritance? They basically explain why Mendel's laws are true. Why Mendel's laws are true. So as we sum this up, a gentleman by the name of Walter Sutton was the first to recognize this, that the behavior of chromosomes, that they segregate during meiosis, the 2 homologous chromosomes segregate, end up in separate nuclei, very similar to what Mendel explained in his law of segregation. Also noted that chromosomes occur in pairs, like Mendel said that for every gene or for every character, there's 2 copies of what determines that character. Each chromosomal pair is separated and delivered separately, singly to gametes. Each gamete only gets 1 of those chromosomes, or gets 1 copy of that, what determines that character. And that separation of one chromosomal pair is independent from the separation of a different pair, explaining Mendel's law of independent assortment. And then in fertilization, one chromosome from each pair is derived from the male and one from the female. Again, explaining Mendel's findings with the law of independent assortment, the law of segregation, and then explaining how offspring are representative of their parents. So he concluded that the alleles for the genes, the alleles for what was making those particular phenotypes, were located on the chromosomes. And he called this that the site on the chromosome must be the site of where you start this process of making proteins. So that wherever this gene was must be where you have your transcription start. All right? Yeah, Joey.
Okay, so for each character,
Yes.
you have in like a diploid cell, you have two traits, either like dominant or dominant recessive or recessive.
Yes.
And then for each one of those traits, you have two copies of it.
No, not necessarily, because if you could have two different traits for the same character. You could have two of the same trait for a particular character. So basically the way it works is Mendel's character is equivalent to a gene on a chromosome, in which there is a place in the code where you write some protein product that's going to shape some feature, right? And so the gene is equivalent to the character. And then for characters, there are different traits, just like for a gene, there are different alleles. And there may be more than two traits possible, right? So if the character is flower color in the pea plant, there just happen to only be two traits, purple and white. But there could technically be more, right? There could be purple, white, pink, blue, you could have more than two different traits. Just like there could be more than two alleles for any specific gene, but each individual can only have two alleles for that gene.
Yeah.
So at most, each individual could have two different traits for the same character, but they're only going to show one if it behaves under normal Mendelian principles, because one trait is going to overpower the other. Yeah.
And then, so the reason that we can end up with 4 different types of gametes.
Well, that's only true if you have two chromosomes. If you have 3 chromosomes, you could generate 8 different types of gametes. If you had 4 chromosomes, you can generate 16 different types of gametes. You can always generate 2 to the number of chromosomes, unique gametes. And that's just factoring in independent assortment, right? So since we have 23 chromosomes, we have 46, but they're 23 pairs. Just if it was only independent assortment, you'd have 2 to the 23rd different gametes that you can make. Now, when you start putting in crossing over, that number gets way, way bigger. 2 to the 23rd is already a big number, but it's way, way bigger than that.
Okay. And that just comes from the 2 steps. First, you separate sister chromatids, and then
Yes.
you separate
No,
homologous.
first you separate homologous chromosomes, then you separate sister
Then you
chromosomes.
separate sister chromosomes.
Yep. And that's the same thing you do as you're putting together a dihybrid cross, right? You set it up, there's 4 different gametes one parent can make, 4 different gametes the other parent can make, and then you mix them. In a trihybrid cross, there are 8 different gametes that each can make. Oh, I also realized I gave you the wrong numbers. When I gave you a tetrahydrid cross and I just kind of spouted it out, it's not 64, it's 81 to 27 to 27 to 27 to 9 to 9 to 9 to 9 to 3 to 3 to 3 to 1. 3 to 3 to 3, 3 to 3 to 3 to 3 to 1. 4 threes. All right, in case you cared. I realized that after class I told you the wrong thing. All right, so which human characters have normal Mendelian inheritance? And this is not an exhaustive suite of all of the characters that behave according to normal Mendelian inheritance patterns, but it's just a few. So here's albinism. And so albinism is actually caused by a point mutation. Okay, you have a point mutation. If you have that point mutation, you're not going to deposit pigment. Doesn't matter all of the other genes responsible for skin color, of how much pigment, what type of pigment, where the pigment is deposited. None of those matter if you have this mutation, because you just cannot deposit pigment. Okay, cannot deposit pigment. So you have albinism, and the albino condition is recessive to normal skin color.
Is that true albinism? Because usually albinism has like pink eyes and they kind of go.
There's variation within even true albinism.
Okay.
Yeah. And then here you have webbed fingers. Webbed fingers is actually a dominant trait. So you have basically two different things that are possible. Either you get, and this is caused by issues in the apoptotic pathway, in case you wondered. You wanted to know some bonus information. The cells here that are supposed to go through programmed cell death to separate these two, they don't do it. Okay, because they actually have a malfunction version of a protein. You're like, well, why doesn't it happen in all the fingers then? It's an excellent question, because that machine is not the only thing responsible for separating the fingers. You can do it without that machine, but it just doesn't work as effectively. Yeah.
Can scientists do that to help separate things?
Oh yeah, you can. Yeah, you can fix this. Now, if you wait until a point where somebody's advanced in age, the first time you find it, it's not worth it, because there's just so many structural adjustments to compensate for it. But yeah, you could do this. I mean, you could do it in the womb if you wanted to. You could do it immediately after birth. You would separate these. Achondroplasia is the most common form of, or one of the most common forms of dwarfism. I don't know if it is the most common. And it again is a dominant trait. And it's a modification in the structural development.
Are these caused by mutations?
Yes. Mm-hmm. Yep. All right. So albinism, recessive to normal skin color, webbed fingers, recessive to normal fingers. No, that's spoken incorrectly. They are dominant to normal fingers. And then achondroplasia, dominant due to abnormal bone growth. All right, so this basically concludes our discussion on normal Mendelian inheritance patterns. And for the rest of chapter 12, we are going to focus on things that do not conform to normal Mendelian patterns. Just like the rest of chapter 12 in your textbook deals with examples of non-Mendelian inheritance, right? Right? Hopefully you've experienced a little bit of that because it's in chapter 12 in your textbook. All right, so we're going to take ourselves a lecture break. And what I want you to do is, working with those around you, I want you to demonstrate how we could predict the offspring ratio if you had more than 2 alleles. Okay? If you had more than 2 alleles for any particular gene, or what you might say, there are more than 2 traits for any particular character. Okay? How would we deal with that kind of a situation? So really what I want you to do is this. The simplest way to do this, or the most straightforward way to do this, would be to cross an individual that, let's say, we'll do them all capital letters. Usually once you get beyond like a 2 allele condition, you don't really use capital and lowercase, because then where do you go from there? Right? You'd use something like this. A1 and A2. So you're going to cross a female that has A1 and A2 with a male that has A1 and A3. Okay? How do we deal with this? What does that look like? How do we handle this? All right? So it's A1 and A3 if the screen's in the way over there. So we're crossing an individual that has A1 and A2. Those are their 2 alleles for this gene. With an individual that has A1 and A3. Yeah.
Are they all dominant?
We're not talking about, let's not deal with phenotypes right now. Because if you start dealing with phenotypes, then it gets a little bit more complicated. Just genotypes. All right? All right, take a minute or two, and working with those around you, come up with a way to work through that.
You know how you can have like, you know how in math they had like 4 exclamation point and then 4 exclamation point.
Wait, you don't just do this?
I bet that's the same thing. Figure out like how many different combinations there are.
There's also depends on the physical location of the gene on the chromosome.
So we're assuming that.
What I mean is when you have in my
You do it in the square.
How do you do a cross?
how do you do
Maybe.
We can set it up like
I think it is a
a
square.
Punnett square.
I mean, because the kids could have A1, A1, or A1, A3, or A2, A1, or A2, A3.
Yeah. This is our A1, A1.
But you couldn't have an A2, A2, A1, A3. A2s has to come
Why
from
not?
a female.
Why can't?
Yeah, it's either 1-1, 1-3, 1-2, or 2.
Because
Yeah.
So I mean, you could set it up like a
I guess you can have.
Punnett square.
Yeah, but the Punnett square doesn't tell you what it looks like. Well, yeah, you said just
Yeah, I guess
yeah.
you're just gonna
So these are potential genotypes.
Uh-huh.
So you could have there's one possible.
All right.
Yeah,
So take a look up here. So you do it the same way you would with a 2 allele situation. You just figure out what kinds of gametes can this individual make. If we have a female that has 2 different alleles, half of her gametes are going to have A1, half of her gametes are going to have A2. Right? We have a male that has A1 and A3, half of his gametes are going to have A1, half of them are going to have A3. Right? And then we fertilize, right? We fertilize the ova, so this is going to have a zygote that has 2 copies of allele 1. This is going to have a copy of allele 1 and a copy of allele 2. Genotypically, it's going to be just like the mom. This individual is going to have a copy of A1 and a copy of A3. Genotypically, going to look just like the dad. And then this one is going to have A2 and A3. So as far as genotypes go, you just execute it the same way you would if you only had 2 alleles for a particular character. But then it gets a little bit interesting as far as what's going to happen with the phenotype. And oftentimes you would have something like this. A1 is dominant to A2, which is dominant to A3. Okay? So A1 is dominant to A2, which is dominant to A3. And so this is kind of cool. If we were like, what's the phenotype of mom? A1, right? She's expressing you only need 1 copy of the dominant. What about the the phenotype of dad? Also A1. And what's the genotype of all 3 of these individuals? A1, but this individual, A2.
So you can't even get an A3.
Something you haven't seen before. No, you can't get an A3 because a mom would have to have A3 since that's the most recessive. But then you get offspring that you've never seen before. And this is even in human characters, this would still be normal Mendelian inheritance. It's a little bit more complicated because we have more than 2 traits. But it still is one is dominant to the other, and we get some phenotype that wasn't present in the parents, right? Coming back up.
So is that how kids can look completely different on their brain?
Part of the reason, yeah. Part of the reason. Another one is mutations, like mutations in the germline. So, I mean, you have on average between 40 and 100, and you're like, well, if it's on average, why can't you just give us a specific number? But between 40 and 100 mutations, neither of your parents had. That you developed, that either developed, or it's a combination of what developed in their germline and also what you've developed during your life. So yeah, so you have 40 to 100 new mutations that your parents didn't have. So, and if those happen in a section that contributes to a phenotype, then that would explain another reason why you could look nothing like your parents.
That only 40 to 100 mutations, well, your DNA is really long. You have
Yeah.
So that's really a very small percentage of your
Right, you have 6 billion. You have 6 billion nucleotides because you have 3 billion in each genome, and you have 2 genomes. So 40 to 100 compared to 6 billion spots, is not it's not significant. That's why there's a low probability you're not going to look like your parents. But it happens. It happens. All right. Oh gosh, I need to ask the question first. So how does incomplete dominance compare with codominance? And so we have 2 things that are very clearly non-Mendelian inheritance patterns. And one is called incomplete dominance, and one is called codominance. Okay, incomplete dominance and codominance. Here you have an example of incomplete dominance. So these are not pea plants with the purple and white flowers. These are snapdragons with red and white flowers. And if these behaved like the pea plants, red should be dominant to white, right? And so if you make a monohybrid, which is happening here, this is a 1-character cross between a true breeding red individual and a true breeding white individual, it should produce an offspring that is red, right? Heterozygous, but showing the red characteristic. It produces an individual that's heterozygous, but you get a blend. You get a mixing of the 2 because this is incompletely dominant. And so red can't completely mask white and instead gets mixed with it and produces pink. And this is kind of the idea behind blended inheritance. And so it's like if that happened, you're like, oh yeah, it's just blended inheritance, right? The dominant individual, which in this case, who knows, we'll say the dominant individual was dad. So dad's characteristics just blended with mom's. And dad didn't give enough of those particles from his cells that make him red to his gamete to make a red individual. He just gave enough to give a pink individual.
Does that happen in humans? Or are there
Oh yeah, there are traits that are incomplete dominance. A lot of traits are incomplete dominance. The problem with humans is we have very few meaningful phenotypes that are controlled by a single gene. And so you have way more complicated factors going on as well. And so here is the monohybrid cross of your heterozygous individuals. Now your heterozygous are pink. They're a mix of the two phenotypes. And then we get our 3-to-1 ratio, but our heterozygous individuals are no longer showing the dominant phenotype. Instead, they are showing a mix. So we get one dominant offspring for every two new phenotype, the heterozygous state, to every one homozygous state. All right? Make sense? I mean, in terms of how it all works out and how the chromosomes behave, it's just like normal Mendelian inheritance. The difference is, what does that mean for the phenotype? What does that mean for the phenotype? All right, so that's incomplete dominance. Codominance is different. The best example of codominance is blood type. And so in blood type, the particular letter refers to antibodies, or it refers to sugars that are on the surface of your red blood cell. And I don't know why we label them A and B, because they have names that are nothing like A and B, maybe for simplicity's sake. So A is a specific sugar on the surface of your red blood cell. B is a different sugar on the surface of your red blood cell. And O is the lack of sugar on the surface of your red blood cells. All right? So in this case, A and B are codominant. So if you have the A allele, you're going to make that sugar, and it's going to be on your red blood cells. If you have B, the B allele, you're also going to make that sugar, and it's also going to be on the surface of your red blood cells. Okay? So that's where you can get 4 different blood types. You can be A-type blood. So you either have one copy, or you either have 2 copies of the A allele, or you have a copy of the A allele and a copy of the O allele. So you have one allele that's saying don't make any sugars. You have another allele that's saying make one type of sugar. Now one type of sugar is dominant to making none. Right? Okay. Same thing with B. For if you have B-type blood, you can have 2 copies of the B allele, or you can have 1 copy of the B allele and 1 copy of the O. The O is saying don't make any sugar. This one's saying make the B sugar. Whether you have 1 copy of that or 2 copies, you're going to put the B sugar on your red blood cells. But if you have A and B, you're going to make both sugars. So those are codominant. If you wanted to use the example previously of the flower, rather than having a pink flower, it would be having a red and white polka dotted flower. Okay, where you're expressing both phenotypes. You're not mixing them. You're not getting a new phenotype. You're just getting both phenotypes.
So let's say I'm a breeder. Let's say I'm like a snake breeder. I'm trying to make a certain morph.
Okay.
How do I know if my trait in one snake and trait in the other snake, how do I know those 2 traits are going to be dominant so that I can get a piebald or a polka dotted snake?
You don't until you start carrying out the breeding and seeing what happens in the offspring. You don't know. I mean, unless somebody sequenced the genome of the snake and somebody has a clue to that. But yeah, you wouldn't know until you start breeding them. And this is what I have to assume happened with the Chihuahua. Right? That they just didn't know what was going to happen, and then until it was too late, right? And it already existed. And then there was nothing you can do. And then they just keep breeding. Sorry if you love Chihuahuas, but and so here's a cross. This is obviously impossible for an individual to have 3 different gametes that they're making with regards to red blood cells, because if they are AB, then they can make 2 different types of gametes, one that has the A allele and one that has the B. This is showing for an entire population. Entire population, there are 3 different types of gametes. One that carries the A, one that carries the B, and one that carries neither. And then you can cross all of these and see what your possibilities are. And notice the only way to get type O blood is to have 2 recessive alleles. And yet it's by far the most common blood type.
Wait, really?
Oh yeah. Oh yeah. It's like, how does that happen? The answer is, I don't know. I don't know. Must be positive selection or negative selection against the blood types, or positive selection for it. And you're like, well, that's kind of weird. Is there any fitness advantage to having O-type blood? Not really. Yeah.
So A and B are co-dominant, but they're both dominant to O?
Yes. A and B are co-dominant, but they are both dominant to O. Yep. Yep. And so to sum this up, how does this compare? Well, the phenotypic ratios in a incomplete dominant situation is 1 to 2 to 1. 2 of the new phenotype that didn't exist in the parents. Hang on just a second. The dominant doesn't completely mask the recessive, and so instead you get a blend, a new phenotype, and then both traits disappear in the F1, not just the recessive. Both traits disappear in F1 in a one-character cross with true breeding individuals. And then the phenotypic ratios in a monohybrid cross with codominance is also 1 to 2 to 1, and this is in terms of giving you if you have A, B, and O. And both alleles are obvious. Neither disappears in the F1, and then blood type is a good example. And so, most of these have more than 2 alleles, and then it adds some complication to these crosses. Yeah, Alyssa.
So O is considered recessive even though it expresses its gene every time?
It doesn't express its gene every time, because if you have an A, it's going to mask the no sugar, right? It's saying no sugar, but if you have another one that says A, you're going to express that A sugar on the surface.
But if O says no sugar, then it has no sugar but also A. So like,
Yeah, but I mean, you could say that same thing about any recessive character. If you have one recessive copy, let's say if you have a heterozygous individual for flower color in pea plants, right? They still have an allele that says white flower, but the flowers are all purple, right? But it's saying white flower, but the purple, whatever that purple pigment, masks the fact that it's saying white flower. So that's how every recessive phenotype works in a heterozygous condition. There's still something they're saying, like, because the white flowers don't deposit the pigment. It's almost exactly like don't make the sugar and put it on the red blood cell surface. But then there's another one saying, no, we're going to deposit the pigment. Just like there's another one saying, no, we're going to put the type A sugar on the surface.
but is it like, is there less A than there would
No.
be was A? Exactly the same?
I don't know if it's exactly the same, but I don't know of any correlation between the whether you are heterozygous A and O versus being homozygous for A and the amount. I don't think there's a connection there. Yeah. Is it exactly the same? Probably not, but every one of your red blood cells probably has a different amount of that sugar anyways.
I was wondering if it was like roughly half.
No, it's certainly not half. Yeah. All right. When you're like, well, then what happens with A and B? Is it half and half? Is there half of each? In some red blood cells, yes, but in some you have more B than A. There's some variation. All right. What is epistasis? What is epistasis? So epistasis, this is the first example where we're dealing with more than one gene. Okay? So in all of our examples so far, even our non-Mendelian patterns of codominance, incomplete dominance, it's still just a single gene. This is more than one gene. And here's an example of epistasis. And so you have 2 different pigment types. One that's light brown and one that's dark brown. One that gives you a black Labrador and one that gives you a chocolate Labrador. Okay, 2 different types of pigment. Black is dominant to brown. Okay, so all of your heterozygous individuals would be black. The only way to get the chocolate is to have homozygous recessive, homozygous recessive. And then, so that's normal Mendelian inheritance. But you have a second gene that controls whether or not pigment is deposited. And therefore, it doesn't matter what the color pigment is, black or brown. If you don't deposit the pigment, you're not depositing brown pigment, and you're going to have a yellow lab, one that lacks pigment in the hair. All right? And so we would say that this gene here, the depositing pigment is epistatic to this gene here, which is pigment type. Because if you can't deposit pigment, it doesn't matter what pigment type you're making. It's not getting deposited. All right? Happens to be the recessive trait is don't deposit pigment. But yeah, you've got two different genes interacting, and one gene can mask the effects of the other. A good example of this in humans is male pattern hair baldness and a widow's peak, right? So widow's peak, you've got, either you have a widow's peak or you don't, but if you have male pattern baldness and you lose your hairline, it doesn't matter whether you make a widow's peak or not, because you don't have any hair. Right? Yeah.
So I heard someone say that male baldness comes from your mom's dad. How do they, technically, if that were true, how do they find things like that?
Well, so the main gene contributing to baldness comes from the X chromosome. And if you are male, you inherit your X chromosome from your mother. Now, she could have gotten her X chromosome that carried that main mutation from her mom or her dad. If she got it from her dad, there's a good indication of what your hair is going to look like where you grew up. But she could have got it from her mom. But then her mom has two X chromosomes, so she could have a normal X chromosome and a bad one, and so she's not going to go bald. And that's why baldness is way less common in women than it is in men, because the main gene contributing to it is on the X chromosome. Women have two and males only have one. Same reason why red-green color blindness is way more common in males than it is in females, because the main gene responsible for it is on the X chromosome. And if you're male, you only have one chance to get it right. If you're female, you have two chances to get it right, because you only need one good copy. All right? So I used to have a widow's peak once upon a time. Not anymore. Yeah.
Wait, but I've never seen a bald lady before.
Oh, you have.
Yeah.
You have certainly, you have certainly seen it. It's easier to hide, right? You just put a hat on, right, like a nice fancy hat, or a wig. Yeah. Or it's like, so the guys when they've got, if they're really bald, and they do the comb over, it looks a little obnoxious. But if you're just partially bald and you have really long hair, you can hide it. Certain hairstyles. But yeah, you've seen it. It's just way less common than it is in men. All right. Oh my goodness. Okay. So here's the cross. And here you have your epistatic gene, which is for the deposit of pigment. All you need is one of the dominant alleles in order to deposit the pigment correctly. So anything with a capital E is going to allow you to deposit the pigment, and then we'll submit to the other one. Okay, so all you need is one capital E. All of these individuals have at least one capital E, so they're depositing the pigment. And then they have at least one capital B, and they're depositing the dominant pigment, which is black. These individuals here, oh gosh, here are the brown. Well, that's really hard to see. Here are the brown individuals. They have at least one capital E, so they are depositing pigment. But they do not have at least one capital B, so they are depositing only the light brown pigment, the chocolate pigment. And then here are these four individuals. They do not have at least one capital E. So they cannot deposit pigment, doesn't matter what color it is. If this one could deposit pigment, what color would it be?
Black.
Black. What about this one?
Black.
This one?
Black.
This one?
Brown.
Brown. Yeah. All right. So what is epistasis? When two genes interact and the genes of one, we call that the epistatic gene, mask the effects of the other. We call that the hypostatic gene. A good example of this, again, genes that determine whether or not the pigment is deposited are often epistatic to the genes for pigment type. An individual cannot deposit pigment, doesn't matter what the color is. Make sense? Right? Doesn't matter that I have the genetic programming for a widow's peak because my hairline has receded too far. I could draw it in like you draw on your eyebrows if you pluck them too thin. That would be really fun. I'm going to do that someday. I was doing colors with my 2-year-old, and I was just asking her, like, what color are your eyes? What color are daddy's eyes? What color are mommy's eyes? My wife has brown eyes. And she said, they're brown, like poop. Because she's like, we're potty training her, so she's just fascinated with poop right now. And I was like, that's right. I said, what color is mommy's hair? It's also brown. She said, it's brown, like poop. And she was getting really excited, so I said, what color is my hair? And she said, orange, on your mouth. Yeah, I guess. And then I was like, what about my hair? And she wouldn't answer it because I think she felt uncomfortable. Like, she didn't want to tell me, like, well, I don't know because there's not very much of it. All right. Oh, gosh, this is fun. Yeah, Rachel. Oh, yeah, sure. You bet. Yeah, Joey.
Just for fun, it's like when people get old and they start having gray hair.
Yeah.
It's just they're losing the ability to produce pigment.
Yeah, some of it is that, like you're losing the ability to deposit pigment. A lot of it is you're not depositing as much pigment. And so you're still depositing pigment, because if you don't deposit pigment at all in your hair, it's blonde. It's blonde. If you go gray or you go white, there's really some really interesting things happening, usually in the patterns of how the pigment's being deposited. And then if you go completely white, there's an issue also in the quality of the cells. So it's not just a pigment issue, it's also an issue in the quality of the cells and how the hair is actually constructed. Because if you look at a hair, you've got the core, you've got the cortex, and then you have the medulla, and you have, what's that outer? Maybe the outer is the cortex. Outer's the cortex, and their medulla, and then the core. I don't know. Anyways, you have 3 different layers to the hair, and so the structure of the hair also helps to determine that as well.
Interesting.
Yeah, because like a polar bear's hair is clear, and somebody who has white hair, their hair is pretty close to clear as well. And the reason why it's looking white is in how light is reflected off of it.
Because white light is like all the light is
Right.
reflected,
Yep.
How about when it's wet, it's translucent?
The polar bear.
Oh, white. Well, my sister had white hair, and when she would take a bath.
Yeah, I mean it's basically it's almost see-through. Yeah. Any other questions?
So what exactly makes a gene recessive or dominant? Is there something like what exactly?
We'll talk about that on Wednesday. So ask that question again on Wednesday. Real quickly, I want to just address one last question, and then we will do our quiz. So why do some character traits form a spectrum? Right, to where it's not like, brown, blonde, brown, blue, webbed fingers, no webbed fingers. Why do some character traits form a spectrum? For instance, like height. And so here's actually a pretty cool figure done at Yale, or at Brigham Young University. Sorry, I saw the Y, got excited for Yale. But Brigham Young, where they took a lab or a class and organized them according to height. And so you can see they built a nice bell-shaped curve. And so we've got height going on. Yeah.
Wait, are you asking us to?
You can answer it if you want.
Is it just because there's lots of different genes involved?
Yes. Yep. Absolutely. And so what you see, according to this class, they were a little bit deceptive in their picture. The range of heights actually looked like this. So it was relatively bell-shaped, but a really weird-looking ugly bell. But you see most of the people fairly close to the average condition. And then as you get further away from the average condition, you have fewer and fewer individuals representing those heights. And so here's an ideal bell-shaped curve. The reason why you have some traits like this that have a continuum is because there are many genes influencing that phenotype. You have a number of genes influencing that phenotype, so it's not quite as simple as tall, short, like in the pea plants, right? You have some tall pea plants, you have some short, and that's normal Mendelian inheritance. This is a little bit more complicated than that. You have several genes that contribute to development and growth. And then something we'll talk about again on Wednesday. We are actually really close to finishing 12. That we'll talk about on Wednesday. But whenever you have this condition, the environment plays a huge role in what you actually get. So you can have the genetics for tall height, but if you are malnourished, you're likely not going to be tall. Right? That makes sense? Or if you are, if you have the genetic predisposition to being short, but you are overnourished and given certain things that actually, cause you to develop some more growth hormones, then you could end up being taller than you should be genetically. All right? Any questions, thoughts, concerns, secrets you want to share? I've never actually offered that one before. Joey?
Do steroids stunt people's growth?
It depends on the type of steroid, and it depends on what you're talking about. But there is the potential in given like anabolic steroids that lead to greater muscle development, that giving those in an early age can arrest skeletal growth because it puts more pressure. The muscles put additional pressure on the bone, and so the bones have to compensate for that additional pressure by growing outward and thickening rather than growing long ways. Yeah, you really don't want to do anything that's going to interfere with normal development. Yeah, Anna. So caffeine's a little bit complicated. I think people